The Promise and the Peril of CRISPR. Welcome back to Techstination. I'm Fred Fishkin. That is the title of a new book from Johns Hopkins University Press. And with us is the editor, physician, and award-winning TV writer and producer, Neal Baer. Hi, Neal.
Hey, how are you?
Terrific. Well, you have worked as a writer and producer on ER and have worked on other shows like Designated Survivor, Under the Dome, Law and Order SVU, and you are a lecturer and co-director of the master's degree program in media medicine and health at Harvard Medical School. You're a busy
Yeah,
guy.
pretty busy. Yeah, it's all good.
And with this book, you are turning your attention to gene editing technology, CRISPR. Tell us about the concept of the book that really is not, it's not just your narrative here.
Right, right. It's a collection of essays. I tie them together with a commentary at the beginning, but I've been interested in CRISPR since I first learned about it maybe 10 years ago, and its prospects for improving health, particularly treating people with really terrible debilitating diseases like sickle cell disease, which CRISPR can cure now. So I put together a book with essays on a range of ethical issues relating to the promise of CRISPR, but also things that may be perilous in the future.
So maybe let's chat about the promise first.
Sure.
You've mentioned one big benefit already.
Right, right. And that is treating sickle cell disease. We can cure it. Several people have been cured. There are 100,000 Black Americans in the United States with this terribly debilitating disease that can cause strokes in children, shorten lifespan, and terrible pain. There are 8 million around the world. So this is a welcome reprieve for this terrible disease. There are other diseases that can be cured by CRISPR now, which is gene editing, like beta thalassemia, and some rare protein folding diseases of the liver, possibly some eye diseases. CRISPR is difficult to deliver so far, say to people who have cystic fibrosis or muscular dystrophy, but it's not as difficult to deliver to the blood because you can inject. And really, what it is a cut and paste mechanism where um, the mutation is identified um, and it is cut by enzymes, and the uh, mutation is replaced by the correct sequence. So it's almost like word processing where you change the letters that are incorrect with the correct letters, and you, in a sense, paste them in.
Tell us a little bit more about the process. And it sounds like it could be, priced very a very pricey thing to undergo here.
It is. Can be between $2 and $3 million to treat one person with sickle cell disease. Yet each year, approximately 200 children are born in New York State with sickle cell disease. It costs $1.6 million to treat them for a lifetime, with about 50,000 out of pocket, not to mention the horrendous pain that comes with sickle cell disease. So, and other problems, as I said, like with stroke. Um, so it's very expensive right now to treat people, and it is very complex, the treatment, because they have to be in the hospital for a number of months to get the treatment. But it's a terrible disease and it can be cured. So it's important to understand that CRISPR, there are two different uh, kinds of CRISPR. There's what we call somatic, and the other is called germline editing. So somatic editing is where we edit the gene that, say, causes a disease, and the person will then not have that disease any longer. In sickle cell disease, the the editing turns on fetal hemoglobin. So when a fetus is in utero, they breathe through a hemoglobin. The blood that carries oxygen in their body is called fetal hemoglobin. And once the baby is born, that turns off genetically, and the hemoglobin, the normal hemoglobin that we have, is what turns on. And so, in people with sickle cell disease, the fetal hemoglobin is turned back on, and it floods the sickled hemoglobin, and that's what um, essentially is what makes people not have the disease anymore. Um, that's somatic, and by somatic, it means it's in the body, but it's not the person who is treated with, say, for sickle cell or beta cell thalassemia, will not pass on the new genes to their offspring. So it's a technique for treating people where they are in their lives with the disease. The other form of CRISPR is what's called germline editing. So the germline is the reproductive line. So Dr. He in China, who, as far as we know, was the first person to treat an embryo, did this by CRISPRing the receptor for HIV. And he did this, he said, because the father of the babies, and I say babies because the embryo split into twins after it was CRISPRed early as an embryo, had HIV, but that's not a reason to make babies resistant because, first of all, we can, if one of the parents has HIV, we can prevent HIV from being transmitted. We can clean the sperm and we can prevent transmission from the mother. So it's not clear exactly why he did that, but we do know that the twins were born, and we're pretty sure a third baby was born using germline technique. Why this is controversial is because you're changing the human germline. In other words, these babies will, if they have children with the changed gene. The people who get treated for sickle cell, if they have children, will not pass that on because it is in their body. It's not in their reproductive cells. But when you're working with an embryo, it will. And so this raises questions about um, evolution and enhancement and changing things at the beginning, and who would be able to afford it, and is that right, and should we be doing that to embryos? Now, it's important to know that we do something in some way similar in an editing way that's a little more general, I guess I might say, which is called PGD, or pre-implantation genetic diagnosis. So what is that? If the parents both carry Tay-Sachs and they want to make sure they don't have a baby with Tay-Sachs, a horrible genetic disease, always fatal, the child will die quite young, 4 years old, suffer. Nothing we can do to stop it. We can look at the fertilized eggs, the embryos, and we can not implant those eggs into the mother so that she won't carry a fetus or then a child and have a child with Tay-Sachs disease. And we can also do amniotic testing, and then the fetus could be aborted in certain states. And so in the United States now. Um, so, there's a question of some people raise, is CRISPR really necessary? Because we can look at genetic diseases and say, we can prevent them by doing IVF and looking at PGD, pre-implantation genetic diagnosis. Now, many, many people can't afford IVF. And so they'll have children with genetic diseases. Some of them are fatal, like Tay-Sachs, adrenoleukodystrophy. Others are fatal later on, like muscular dystrophy. possibly CF, though people are living longer, and some are not fatal, like albinism. So where do we draw the line in thinking about, what we should stop or prevent and what is okay? And one person might decide that they want to have a child with Down syndrome. Another person might not. Um, so CRISPR is a way in the future, more likely than now, to possibly enhance. Um, and we have to know more about the specific qualities that the genes have. There's no one gene for intelligence. There's not one gene for eye color, but there are mutations for for pain. So for instance, there are children born who don't feel pain. And I heard a story recently about a kid who jumped off a 4-story building to prove to his friends that it didn't hurt. Well, it killed him, but he had broken bones in the past. Purportedly, Russia and China are doing research to develop soldiers that wouldn't feel pain. They could use CRISPR and make that mutation, or to sleep less, or to have less fear. So, should we be doing germline editing um, to enhance or make soldiers? The other point that I want to raise is that around 2015, and this is in our book, um, the scientific academies pretty much agreed that we shouldn't do germline editing on embryos. We shouldn't change human beings that way. And then Dr. Hood did his work, and 2 babies were born. And scientists changed their tune a little bit. And we write about this and said, well, maybe there are times when germline editing is okay. And they use an example that is interesting that only CRISPR can do. And what is that? And that is, if 2 parents, the mother and the father, are both deaf and they have a genetic deafness, then the chances, depending on that genetic syndrome, they'll never have a child who can hear. Just can't happen. So a Russian scientist has said it's not fair for those parents not to have a child who can hear if they want their child to hear. And we should be doing CRISPR germline editing on that embryo and change the gene so that they will be able to hear. We have a piece in our book by Carol Padden, who's a professor at UC San Diego, who is deaf, and she argues that deafness is human variation, and it's not pathological for her, and we shouldn't just jump at the chance to start CRISPRing babies who might be born deaf. And this raises the question of the slippery slope: is where do we stop? Do we do we you know do we treat children who are all deaf? Do we treat children with albinism? We have a really wonderful piece by a physician from UC San Francisco, Ethan Weiss, who talks about his child born with albinism. If they had known, he says, that their child would be born with albinism, um, he and his wife probably would have decided to abort the fetus. But knowing now the child they have, they couldn't imagine not having that child. So we get into these very complex ethical dilemmas. What's knowable in the future isn't knowable necessarily. And we don't know really what CRISPR can do. It's like um, the
I mean,
promise of
is
plastic.
there potential that vision could be enhanced or hearing could be enhanced, that we'd be super beings of some sort?
I think there's potential if we know where the genes are, where there are super smellers, sniffers, people who smell more things than other people. There are people who hear, at wider ranges. So that's conceivable.
So when you think about laws, regulations, how can they possibly be enforced? I mean, you mentioned, okay, we might have a law against it, but in China, it's go.
Well, it wasn't go. He was put in prison because the outrage was strong. But one of our essays, the last one in the book, talks about how Doctor Who was encouraged by scientists in the United States possibly to do what he did. Ben Hurlbut, the writer, who's a professor at the university or at Arizona State University, was at these meetings. One of the professors, he quotes one of the professors saying, "Don't hire me number one, hire me number five when the controversy is over." And don't you want to be like the fellow who invented IVF? He didn't do it the traditional way. He announced it in the newspapers, and he ultimately won a Nobel Prize. So there's some argument in this piece that Doctor Who was kind of pushed in a way to do some things that they wouldn't do here because it is illegal everywhere. But there's no real enforcement. Researchers have told me that they should be left alone to enforce it on their own. How they would do that, I don't know. By making the rogue scientists pariahs. Well, that's fine, but once they've done it, they've done it. There are things that can be done. We do have a chapter on oversight and legal issues. Um, there are companies that manufacture pieces of DNA that scientists use and purchase to do their work, and these companies are encouraged to keep track of what they're selling and to whom, because we know, for instance, that we know what the DNA makeup of smallpox is and Ebola and things like that. So if somebody's buying pieces that can be put together to do that, we need to know. So we need to have some oversight. We need to talk about this. Have you ever heard a politician talk about CRISPR? They talk about AI, they talk about global warming, they talk about plastic and nanoplastic. They talk about climate change, but they don't talk about CRISPR.
And the
So
potential for altering, I guess, life as we know it is there.
yes, and we already do alter life. Of course, we do hybridization of animals and plants, and we do transhuman alterations where we change organs in pigs and then transplant them into human beings in Boston now. Um, and there are questions. We have a chapter in our book about animals and CRISPR, like what happens if we start to make can we make animals sentient? Can they start to feel in a way that humans feel? How do we think about these things? Should we just be doing it? um, and just say, well, let science, lead the way. That's what Ben Hurlbut argues, is that science has led the way in terms of ethics. They say we shouldn't do it. Someone does it. Then they say, okay, maybe it's okay. Here are some examples of what would be okay. And ethics follows the science, as opposed to the ethics saying there should be a moratorium. We don't know enough. And then there are issues of off-target results and things like that, where their CRISPR is getting better and better, but you can still have off-target mutations, let's say. So a lot of complexity and a lot of promise. First half of our title, promise and peril. The first chapter in our book talks about bioweapons in CRISPR. And, some scientists will argue, well, you can make bioweapons without CRISPR. And yeah, of course that's true. But it might be easier to make it with CRISPR by manipulating, let's say, H5N1 and making it more transmissible. I don't think we want a rogue scientist to do that. How do we have oversight? Just trust that people will behave in humane ways? Kind of worries me a bit. That's why the first line in the book is my chapter, which says, CRISPR keeps me up at night.
It'll keep me up at night too, reading, reading and thinking. The book again, The Promise and the Peril of CRISPR. Neal Baer, thank you so much for spending time with us.
Oh, thank you for having me. It's my pleasure.